Molecular basis of inheritance, part 2, NCRT based single sentence questions. Let's start. During dash and a dash, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. Process which require transfer of genetic information from polymer of nucleotides to synthesize a polymer of amino acids. Translation. Neither does any complementarity exist between nucleotides and amino acid, nor could any be drawn theoretically. True or false? True. Insertion or deletion of three or its multiple bases, insert or delete in one or multiple codon, hence one or multiple amino acid, and reading frame remains unaltered from that point onwards. True. Change in nucleic acid, genetic material were responsible for change in dash in protein, amino acids. During synthesis of proteins, dash direct the sequence of amino acid, genetic code. Further proposition and deciphering of genetic code require involvement of scientists of various disciplines like physicists, organic chemists, biochemists, and geneticists. Physicists argued that since there are only 4 bases and if they have to code for 20 amino acids, the code should constitute a combination of bases. George Gamow. Scientists who developed chemical method which was instrumental in synthesizing RNA molecule with defined combinations of bases. Har Gobind Khorana. Scientists to develop chemical method which was instrumental in synthesizing RNA molecule with defined combination of bases. Har Gobind Khorana. Scientists who suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. George Gamow. Scientists who developed cell-free system for protein synthesis helped the code to be deciphered. Marshall Nirenberg. Scientists to develop cell-free system for protein synthesis helped the code to be deciphered. Marshall Nirenberg. Scientists who made a bold proposition which a permutation and combination of 4 to the power of 3, that 4 into 4 into 4, would generate 64 codons, generating many more codons than required. Scientist George Gamow. RNA molecules with defined combinations of bases are homopolymers or heteropolymers or copolymer. Homopolymer or copolymer. RNA molecule with defined combinations of bases are both homopolymer and copolymer. Enzyme which is helpful in polymerizing RNA with defined sequences in a template independent manner. Severo-Okoya enzyme. Enzyme which is helpful in polymerizing RNA with defined sequences in a template independent manner. Severo-Okoya enzyme. Codon is triplet. Enzyme used in enzymatic synthesis of RNA. Severo-Okoya enzyme. Enzyme used in enzymatic synthesis of RNA. Severo-Okoya enzyme. Number of codon which code for amino acids. 61 out of 64 which code for amino acids are 61. Severo-Okoya enzyme also called as polynucleotide phosphorylase enzyme. Polynucleotide phosphorylase enzyme. Number of codon which act as stop codon. 3. Number of sense codons. Number of sense codons 61. Number of nonsense codons 3. Number of nonsense codons 3. Codons which code for amino acids are called sense codons. Codons which code for amino acids are called sense codons. Codons which do not code for amino acid and doing other activities are called nonsense codon. Codon which do not code for amino acid and doing other activities are called nonsense codon. Some amino acids are coded by more than one codon. Hence the code is here answer we have. Some amino acid coded by more than one codon. Hence the code is degenerate. The codon is read in mRNA in a contiguous fashion. There are no punctuations, hence the code is commaless. For example, from bacteria to human, UUU would code for phenylalanine. Hence the codon is universal. Codon is universal. Codon is commaless. Codon is degenerate. Codon with dual function. Codon with dual function AUG. Codon with dual function AUG. Exceptions in universality of codons are seen in mitochondria and some protozoans. We have exceptions for universality of codons. Codon which code for phenylalanine. Codon which code for phenylalanine UUU. Codon which code for methionine AUG. Codon which code for methionine AUG. Initiator codon is AUG. Terminator codons are UAA, UAG, UGA. Terminator codons are UAA, UAG, UGA. Terminator codon which is also called umber UAG. Terminator codon which is also called ochre UAA. Terminator codon which is also called opal UGA. Codon which code for amino acid and also act as a initiator AUG. Start and stop codons respectively. Start codon, initiator codon is AUG and terminator codon. Stop codons are said to be UAA, UAG and UGA. Number, occur, open. The following is a sequence of nucleic acid nucleotides in mRNA. Predict the sequence of amino acid coded by it. Predict the amino acid coded by it. For AUG, the code is methionine. And for UUU and UUC and UUU, the code is phenylalanine. Following is a sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. Methionine, phenylalanine, phenylalanine, phenylalanine. So that we can say like this: AUG, UUU, UUU. As we said, there is no punctuation marks. We have to draw right like this: UUU, UUU, like this. Or you can add UUC also. The relationship between genes and DNA are best understood by relationship between genes and DNA are best understood by mutation studies. Effect of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function, loss or gain of a function. A change in single base pair is considered as point mutation. Change in a single base pair is considered as point mutation. Classical example for point mutation is sickle cell anemia. Classical example for point mutation is sickle cell anemia. Protein in the hemoglobin is globin. Number of polypeptide chains in the globin 4. Polypeptide chains of globin are alpha, 2 alpha chains and 2 beta chains. Alpha polypeptide chains are represented by alpha genes. Beta polypeptide chains are represented by beta genes. Change in a single base pair in the gene for beta globin chain that results in a change of amino acid residue glutamate to valine. It results into a diseased condition called valine and it results in a diseased condition called sickle cell anemia. Gene sickle cell anemia. Glutamic acid replaced by valine. Consider a statement that is made up of the following words, each having 3 letters like genetic code. Recognize the point mutation as insertion or deletion. Insertion or deletion. We have to say it as frame shift insertion. Frame shift insertion. Okay. Consider a statement that is made up of the following words, each having 3 letters like genetic code, that recognize the point mutation as insertion or deletion. Here it is deletion. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift. Such mutations are referred as frame shift insertion or deletion. Frame shift insertion or deletion mutations. Insertion or deletion of 3 or its multiple bases. Insertion or deletion of 3 or its multiple bases. Dash or dash in one or multiple dash in hence one or multiple dash and reading frame remains unaltered from that point onward. Insertion or deletion of 3 are its multiple bases: insert, or delete in one or multiple codons, hence one or multiple amino acids. And reading frame remains unaltered from that point onwards. From the very beginning, of the proposition of code, it was clear that to Francis Crick that there has to be a mechanism to read the code and also to link it to the amino acid. From the very beginning of the proposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also link it to the amino acids. Yes, true or false? True. Amino acids have no structural specialities. Amino acids have no structural specialities to read the code uniquely. True. He postulated the presence of an adapter molecule that would on one hand read the code and on other hand would bind to specific amino acid. He, who is he? Francis Crick. He postulated presence of an adapter molecule that would on one hand and read the code on other hand would bind to specific amino acid. Adapter molecule is the he adapt Francis Crick, here he Francis Crick as we said, and adapter molecule is tRNA. Adapter molecule is tRNA. tRNA was known before the genetic code was postulated. True or false? True. sRNA is also called tRNA Soluble RNA is tRNA. tRNA role as adapter molecule was postulated before genetic code. True or false? tRNA role as adapter molecule was postulated before genetic code. No, false. The tRNA, then called sRNA, soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. True. Its role as an adapter molecule was assigned much later. True. tRNA has an dash that has bases complementary to the code. Anticodon loop. Anticodon loop. If mRNA has AUG codon, anticodon loop on tRNA is UAC. UAC. Portion of tRNA which can bind with amino acid. Portion of tRNA which can bind with amino acid. Amino acid acceptor end. Amino acid acceptor end. If mRNA has a codon UUU, anticodon on tRNA at an anticodon loop, an amino acid on tRNA at amino acid acceptor end. Anticodon is said to be AAA and amino acid phenylalanine. Phenylalanine. For initiation, there is a specific tRNA required and that is called that is called initiator tRNA. Initiator tRNA. tRNA for stop codons is tRNA for stop codon. There are no tRNAs. There are no tRNAs for stop codons. Stop codons. There are no tRNAs. tRNA looks like a clover leaf, clover leaf like structure. In actual structure, tRNA is a compact molecule. tRNA is a compact molecule that which looks like inverted L shape, inverted L shape molecules. So secondary structure of tRNA has been depicted that looks like secondary structure of tRNA has been depicted that looks like clover leaf. Polymerization of amino acid to form a polypeptide to form mRNA is called translation. Polymerization of amino acids to form a polypeptide from mRNA is called translation. The order and sequence of amino acids are defined by the sequences of bases in the mRNA. Bases in the mRNA. Amino acids are joined by a bond which is known as a peptide bond. It is known as a peptide bond. Formation of peptide bond require energy. Amino acids in the first phase of translation itself activated in the presence of ATP and linked to its cognate tRNA process is called charging of tRNA or amino acylation of tRNA. Charging of tRNA or amino acylation of tRNA. Charging of tRNA or amino acylation of tRNA. Charging of tRNA or amino acylation of tRNA is specific or non-specific. Specific. If two charged tRNAs are brought close enough for the formation of dash bond, peptide bond between them would be favored energetically. Here it is. Peptide bond. If two charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. Charging of tRNA is also called amino acylation. Amino acylation of tRNA. Amino acylation of tRNA. tRNA are amino acid specific or non-specific? tRNA are amino acid specific. Charging of tRNA means activation of amino acids and linking with their specific RNA, specific tRNA. Activation of amino acids and linking with their specific tRNA. Presence of a dash would enhance the rate of peptide bond formation. Presence of a catalyst would enhance the rate of peptide bond formation. Cellular factory responsible for synthesizing proteins is the ribosome. Cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of dash and about dash different protein. Ribosome consists of consists of structural RNAs and about 80 different types of proteins. The ribosome consists of structural RNAs and about 80 different proteins. Ribosome in its inactive state, it exists as two subunits. The two subunits of ribosome are larger subunit and smaller subunit. Subunit of ribosome which first encounters mRNA, smaller subunit. Subunit of ribosome which first encounter mRNA is smaller subunit. The process of translation of the mRNA to protein begins when the dash encounters an mRNA, that is smaller subunit. There are two sites in the dash subunit for subsequent amino acid to bind and thus be close enough to each other for the formation of a peptide bond, that is larger subunit. Two sites are there in larger subunit. Act as a catalyst for the formation of peptide bond. Act as a catalyst for the formation of peptide bond. Ribosome. Act as a catalyst for the formation of peptide bond. Ribosome. Type of RNA which act as a catalyst for the formation of peptide bond in bacteria, that is 23S RNA. 23S RNA, that is rRNA. 23S rRNA. Ribosome acts as catalyst in bacteria. Ribozyme act as catalyst in bacteria. One and only enzyme which is not a protein, ribozyme. What is ribozyme then? Ribozyme is an enzyme but not a protein. Ribozyme is 23S rRNA of bacteria. Ribozyme is catalyst for the formation of peptide bond during translation process. Peptide bond during translation process. A translational unit in mRNA is a sequence of RNA that is flanked by the start codon. Start codon like AUG, start codon AUG, and the stop codons like UAA, UAG, UGA, and codes for a polypeptide. That's the story. UTRs means untranslated regions. UTRs means untranslated region. Additional sequences of mRNA are called as UTRs, untranslated regions. And mRNA also has some additional sequences that are not translated and are referred as untranslated regions. Nothing but we call UTRs. Untranslated regions are UTRs. UTRs represent at 5 end or 3 end? Both ends. UTRs required for efficient translation process. UTRs required for efficient translation process. Location of UTRs at 5 end of mRNA. Location of UTRs at 5 end of mRNA, that is before the start codon, before AUG. Location of UTRs at 3 end of mRNA, that is after stop codon. Before start codon and after stop codon, we find untranslated regions like this. Let's do a little bigger. So this is UTR, untranslated region, of before initiator, after terminator codon, along with 5 capping and polyadenylation. So arrange the sequence capping 5 UTR, protein coding sequences, terminator codon, and 3 UTR, poly A tail in order. G initiator codon and terminator codon. We should start the story with 5 capping. First, it will come 5 capping. Next, after 5 capping, what you can see? UTR, 5 end of UTRs. You can see here 5 end of UTRs. Okay. What next? After 5 end of UTR, we'll find initiator codon. AUG is the general initiator codon. Initiator codon, you can see. Next, what next? The protein coding sequences, whatever present, whatever the protein coding sequences, where it is, here it is, protein coding sequences. Okay, what next after protein coding sequences? Terminator codon will come. Terminator codon. After terminator codon, we can see stop codon or terminator codon, whatever that is next. After that, 3 UTR, untranslated region. As we said, after stop codon, we'll find untranslated region, and after that only you'll find our so-called polyadenylation, poly A tail, which is there in eukaryotic cells, that 3 UTR, and finally the polyadenylation, poly A tail in order. So this is the complete order. In a particular mRNA, we start with 5 capping and 5 UTR, then initiator codon and protein coding sequences, terminator codon, 3 and untranslated region, and then polyadenylation, eukaryotic mRNA. Okay, then next, moving on to the, for initiation, the ribosome binds to the mRNA 8. For initiation, the ribosome binds to the mRNA 8 start codon. mRNA 8 start codon, that is in general AUG. Okay, then that is recognized only by initiator tRNA. It's a special tRNA, that is initiator tRNA. Initiator tRNA. The ribosome proceeds to the dash phase of protein synthesis. Ribosome proceeds to the elongation phase of protein synthesis. Ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA and T codon. During this stage, during the translation phase, complexes composed of an amino acid linked to tRNA and sequentially bind to the appropriate codon in mRNA by forming complementary bases with the tRNA and T codon during translation process when elongation is happening. The ribosome moves from codon to codon along. Ribosome moves from codon to codon along the sequences of the amino acids are added one by 1 translated into DNA sequences dictated by mRNA. Ribosome moves from codon to codon along the sequences. Amino acids are added one by one, translated into DNA sequence, translated into, here it is, translated into amino acids, into amino acid sequences dictated by DNA and represented by mRNA. DNA send the messenger. Based on that, the translation is happening and the amino acid sequences are going to be in order. At the end of translation, a dash factor binds to the stop codon. The release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Complete polypeptide from the ribosome. Okay. Then, if mRNA has codon AUG, anticodon base sequence present on the specific tRNA is if mRNA has codon AUG, anticodon base sequence present on the specific tRNA is UAC. If mRNA has codon UUU, anticodon base sequence present on the specific tRNA is AAA. Regulation of gene expression refers to a very broad term that may occur at various levels. True or false? It's true. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. It can be regulated at several levels. True or false? True. In eukaryotes, the regulation could be exerted at transcriptional level. That means formation of primary transcript. In eukaryote, the regulation could be exerted at the 2 processing level. That is regulation of splicing. Regulation of splicing. In eukaryotes, the regulation could be exerted at number 3 transport of mRNA from nucleus to the cytoplasm. From nucleus to the cytoplasm. In eukaryotes, the regulation could be exerted at transcriptional level or processing level or transport level or translational level. In all the levels. The genes in a cell are expressed to perform a particular function or a set of functions. True or false? True. Enzyme beta galactosidase is synthesized by Escherichia coli. Enzyme beta galactosidase is used to catalyze the hydrolysis of a disaccharide lactose into monosaccharides like glucose and galactose. Enzyme beta galactosidase synthesized by Escherichia coli, and the beta-galactosidase is used to catalyze hydrolysis of a disaccharide lactose into glucose and galactose. Enzyme required for the digestion of lactose in E. coli is enzyme required for the digestion of lactose is beta galactosidase. Beta galactosidase, in general terms, we say lactase. Gene responsible for the production of beta galactosidase in E. coli is Z gene. Functioning status of Z gene and group when lactose available, active or inactive? Active. Functioning status of Z gene and group when lactose not available to E. coli, active or inactive? Inactive. Functioning of a gene depends on the metabolic, physiological, or environmental conditions that regulate the expression of genes. True or false? True. Irrespective of requirement, gene actively produce their products. True or false? False. The development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several sets of genes. True or false? True. In prokaryotes, control of the rate of transcriptional initiation is the site for predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory protein, which affect its ability to recognize the start sites. These regulatory proteins can act. These regulatory proteins can act both positively, like activator, positively, that means activators, or negatively, that means repressors. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory protein, which affect its ability to recognize start sites. These regulatory proteins can act as positively or negatively. Yes, both positively or negatively, activator or repressor. The accessibility of a promoter region of prokaryote, the accessibility of promoter region of prokaryotic DNA is in many cases regulated by the interaction of proteins with a sequences termed operator. Sequences termed as operator. Operator region is adjacent to the dash elements in most operon, adjacent to the promoter. In most the sequences of the operator bind a repressor protein, bind with the repressor protein. Each operon has its specific operator. True or false? True. Each operon has its specific repressor. True or false? Yes, true. Lac operator is present only in the lac operon and it interacts specifically with lac repressor only. True or false? True. Elucidation of the lac operon was also a result of close association between a geneticist and a biochemist. Geneticist Francois Jacob and biochemist Jacques Monod. Jacques Monod. They were the first elucidate a transcriptionally regulated system. Same old Jacob and Monod. Jacob and Monod. In lac operon, lac refers lactose. In lac operon, operon means group of genes. In lac operon, polycistronic structural genes are Z, Y, A. Z, Y, A. Like lac operon, other operon in bacteria as other operons like tryptophan operon, we can see that ara operon, his operon, histidine operon, val operon, valin operon, likewise. The lac operon consists of a dash gene. The lac operon consists of a inhibitor, B Z, Y, A. We call them as structural genes. Inhibitor is there and structural genes like Z, Y, A. In lac operon, I refers inhibitor. In lac operon, structural genes are Z, Y, A. In lac operon, IgG code for repressor of the lac operon. Repressor, inhibitor protein. In lac operon, Z gene code for enzyme beta galactosidase. Z gene code for enzyme beta galactosidase. In lac operon, Y gene code for enzyme permease. In lac operon, A gene code for A gene code for transacetylase. Transacetylase. In lac operon, A gene code for enzyme transacetylase. All these are enzymes. You should know them. Enzyme permease, which increases the permeability of intake of lactose into the bacterial cell. Enzyme transacetylase. Enzyme which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units called glucose and galactose is beta galactosidase. Enzyme beta galactosidase or That is from Z gene, isn't it? Enzyme which increases the permeability of the cell membrane, permeability of the cell to the beta galactosidase, that is permease, which is produced from Y gene. All the three gene products in lac operon are required for metabolism of lactose metabolism. They participate in lactose metabolism. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. True or false? True. In I gene, I refers inducer. True or false? False. I means inhibitor. Substrate of the enzyme beta galactosidase is lactose itself. Substrate for the enzyme beta galactosidase activation lactose itself. It regulates the switching on and off of the operon, that is lactose. That it regulates switching on and off of the operon, hence it is termed as lactose is a regulatory and it is termed as inducer. In E. coli, the absence of a preferred carbon source such as glucose, preferred carbon source is glucose. If lactose is provided in the growth medium of the bacteria, the lactose is transported in the cells through the action of an enzyme called enzyme permease, which is produced from the transcription and translation of Y gene. A very low level of expression of lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cells. True or false? A very low level of expression of lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cell. True or false? True. The dash of the operon is synthesized all the time constitutively is the dash of the operon, that is repressor. Repressor protein is produced from I gene. Repressor protein is produced from I gene. If repressor protein binds to operator region of the operon, it prevents the movement of RNA polymerase. It prevents the movement of RNA polymerase enzyme towards towards the structural gene and prevent transcription of structural genes. Prevents transcription of structural genes. Okay. So what we are saying here in repressor protein binds to operator region of the operon. It prevents the movement of RNA polymerase enzyme toward the structural gene and prevent the transcription of structural genes. Operator is in off mode when operator bind with operator is in off mode when operator bind with repressor protein. Operator is on mode when operator detach with repressor protein. To make detachment of repressor with operator, it is required. Who is required? Inducer. Inducer, that is nothing but lactose or allolactose. Inducer here is lactose or allolactose. Repressor protein is produced from the gene. Repressor protein is produced from the gene, that is a special gene, I gene or inhibitor gene, we can see. Inducer, which is the lactose or allolactose, binds to operator Operator detached from repressor. Operator is on. It allows the RNA polymerase to activate structural genes for transcription. Transcribed structural gene form single mRNA. The single mRNA from the three genes is called polycistronic RNA, mRNA, polycistronic mRNA, which is formed by the transcription of Z, Y, A. All the three is going to form only one mRNA, that is called polycistronic mRNA. To stop structural gene functioning, need to stop RNA polymerase activity. RNA polymerase activity will be stopped when operator is off. How operator can be switched off? How does the operator can be switched off? When operator binds, when operator binds with repressor protein, operator binds with repressor protein again. The dash of the operon is synthesized all the time constitutively from the I gene. Repressor. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. If the presence of an inducer, such as, if the presence of an inducer, such as, with the inducer lactose or allolactose, lactose or allolactose is the inducer. The repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. This allows RNA polymerase access to the promoter and transcription proceeds. That is detachment of operator with repressor. Essentially, regulation of lac operon can also be visualized as a regulation of enzyme synthesis by its substrate. Glucose or galactose cannot act as inducers for lac operon. True or false? True. Glucose or galactose can act as inducer for lac operon. False. For lac operon, inducer can be lactose or allolactose. For lac operon, inducer can be lactose or allolactose. True. Regulation of lac operon by repressor is referred to as negative regulation. Lac operon is under control of positive regulation as well. True or false? True. Can you think for how long the lac operon would be expressed in the presence of lactose? The lactose operon expresses as long as the lactose present, as long as the lactose is present. When all lactose is converted, when all lactose is converted into glucose and galactose, the reaction stops. Lactoseperon expresses as long as the lactose is present. When all lactose is converted into glucose and the galactose, the reaction stops. Genetic makeup of an organism or an individual lies is in the DNA sequences. Lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. With the establishment of genetic engineering techniques, with the establishments of genetic engineering techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. In the year 1990. A mega project aimed to find out the complete DNA sequence of human genome is called Human Genome Project. The month and year which international efforts has begun on HGP, October 1990. Time period taken for the completion of HGP, 13 years. Trust of UK, which became a major partner during the early years of HGP. Welcome Trust. Organizations which coordinated for HGP, that is US Department of Energy, and National Institute of Health. Countries who made additional contribution for HGP, that is Japan, France, Germany, and China, others. HGP is almost completed by this year, 2003. HGP was closely associated with the development of a new area in biology called bioinformatics. Besides providing clues to understanding the human biology, learning about the non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied toward solving challenges in healthcare. Solving challenges in healthcare and agriculture, that we can solve the challenges of the healthcare, agriculture, energy production, and environmental remediation. Genomes of many non-human models organisms which have also been sequenced along with human genome, they are they are a unicellular prokaryote. A unicellular prokaryote is that a unicellular prokaryote in Escherichia coli, and a multicellular eukaryote, that is yeast, a free living non-pathogenic nematode, Cenorhabditis elegans. Cenorhabditis elegans. Cenorhabditis elegans is a non-pathogenic nematode, a free living non-pathogenic nematode. Insect. Insect is Drosophila melanogaster. Drosophila melanogaster. Then plants, the plants like rice and Arabidopsis thaliana. Arabidopsis. So unicellular prokaryote Escherichia coli, multicellular eukaryote yeast, the free-living non-pathogenic nematode Caenorhabditis elegans, an insect Drosophila melanogaster, plants like rice and Arabidopsis thaliana. A set of DNA instructions in a cell is called a set of DNA instructions in a cell is called genome. Whole hereditary information encoded in DNA is called genome. Organisms, complete set of deoxyribonucleic acid molecules is called genome. Haploid set of chromosomes is called genome. Haploid set of chromosomes in a gamete or a microorganism or in each cell of multicellular organism is called genome. The complete set of gene or a genetic material present in a cell or organism is called genome. Number of chromosomes in the genome of male human 24. Number of chromosomes in the genome of female human 23. In male we have Y chromosome. Why one chromosome need to be studied additional in male when compared to female? That's what we are saying. In male there is Y chromosome which is present. Y chromosome is present which is not completely similar to the X chromosome. That's why we have to read additional chromosome in case of human being, a Y chromosome in case of male human being. Next, a chemical compound that contain the genetic instruction needed to develop the direct activities of the every organism present in the form of chromosomes is called DNA. Genetic makeup of an organism or an individual lies in the form of DNA. Approximate number of genes which HGP aimed to identify 20,002. They approximated calculation they made in the initial 20,000 to 25,000. Number of sequences of the chemical base pair which HGP is aimed to determine 3 billion chemical base pairs. What is ELSI with respect to the goals of HGP? ELSI, that is addressing the ethical, legal and social issues. Ethical, legal, social issues that may arise during the project from the project. The approach of HGP, which focused on identifying all the genes that expressed as RNA, is referred to as expressed sequential tags. The approach of HGP, which focused on identifying all the genes that expressed as RNA, is referred to as expressed sequential tags. Very, very important. The blind approach of sequencing the whole set of genome that continued all the coding and non-coding sequence and later assigning different regions in the sequence it functions is referred as sequence annotation. Sequence annotation. These two are very important. That the first, the approach of HGP, which focused on identifying all the genes that expressed as RNA, is referred to as expressed sequential tags. The blind approach of sequencing the whole set of genome. Here, protein coding genes. Here, whole set of genome that contain all the coding and non-coding sequence and later assigning different regions in the sequence with function is referred to as sequence annotation. The process of determining the exact order of the 3 billion paired chemical building blocks that make up the DNA is called. That is DNA sequencing. What is the first step in the process of DNA sequencing? Total DNA from a cell is total DNA from a cell is isolated and converted into random fragments of relatively smaller size. Total DNA from a cell is isolated and converted into random fragments of relatively smaller size. The nitrogenous bases of DNA are adenine, thymine, guanine, cytosine. Adenine, thymine, guanine, cytosine. Methods followed for the amplification of DNA fragment which are used for sequencing the bases. Cloned in a suitable host, that can be cloned in a suitable host using specialized vectors. The commonly used hosts for the cloning of DNA fragments of human genome are commonly used are bacterial artificial chromosome, yeast artificial chromosome. That bacterial artificial chromosome, BAC, bacterial artificial chromosome, and yeast artificial chromosome. Methods used for the sequencing of DNA fragments. Methods used for the sequencing of DNA fragments is automated DNA sequences. The latest method of DNA sequencing, which is used even for the sequencing of longer fragments and replaced the traditional sequencing method, that is shotgun sequencing. Shotgun sequencing, which uses which is used in which uses supercomputers. Automated DNA sequences work on the principle of a method developed by which is developed by Frederick Sanger. Frederick Sanger is a scientist. Automated DNA sequences work on the principle of method which is developed by Frederick Sanger. Arrangement of DNA sequences was humanly not possible. Then this problem nowadays this problem is solved by specialized. Now this problem is solved by specialized computer based programs were developed and then sequences were subsequently annotated that and were assigned to each chromosome. Specialized computer based programs were developed and then sequences were subsequently annotated and were assigned to each chromosome. haploid set of chromosomes of human which are made up of nucleotide base pairs is called genome. Answer the question with respect to human genome. The total number of nucleotide base pairs 3164.7 million base pairs, or we say 3 billion base pairs. Average number of base pairs in a gene 3000. Largest known gene with 2.4 million base pairs, that is dystrophin. Do all the genes have same number of base pairs? No. Vary greatly in size and number. Total number of genes estimated are 30,000. Previous estimation on total number of genes in human 80,000 to 1,40,000, but that much number is not there, only 30,000. Percentage of nucleated bases expected as exactly same in all people 99.9%. Percentage of genes discovered whose function is unknown, that is 50%. Percentage of the genome who code for proteins, that is less than 2%. Sequences which make up very large portion of human genome, that is repeated sequences. Chromosome with highest number of genes. First chromosome, which has 2968 genes are present on the first chromosome. Chromosome with lowest number of genes, that is Y chromosome with only 231 genes. Number of genes on first chromosome, that is 2968 genes. Number of genes on the loci of Y chromosome, that is least of all, that is 231. Number of location where single base DNA differences are present, that is about 1.4 million locations are having a single base difference. Single nucleotide polymorphs, we call them as, are pronounced as SNPs. They have only single base differences. We call them as SNPs, single nucleotide polymorph. DNA sequence variation that occur when a single nucleotide in the genome sequence is altered are called as SNP, single nucleotide base differences, or single nucleotide polymorphs, we can say. This information promises to revolutionize the process of finding of chromosomal location or for disease associated sequences and tracing human history, that is information on SNPs. This information promises to revolutionize the process of finding chromosomal locations for disease associated sequences and tracing human history. That is information on SNPs. Stretches of DNA that repeated many times, sometimes 100 to 1000 times, are called as repetitive sequences. They are thought to have no coding function, but they shed light on chromosome structure dynamics on evolution. That is repetitive sequences. Cost of sequencing required estimated cost in the beginning for one base pair in US dollars, that is $3 per base pair. Total estimated cost of the project approximately 9 billion US dollars. 9 billion US dollars. If the obtained sequences were to be stored in typed form in books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, 1,000 pages, then such books, then 3,300 such books would be required to store the information of DNA sequence from a single human cell. Enormous amount of data expected to be generated also necessitated the use of high speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of new area in biology is called bioinformatics. Deriving the meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. This enormous task will require This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having HG sequences may well be enabling a radical and new approach to the biological research. True or false? It's true. In the past, researchers studied one or a few genes at a time. True. In the past, researchers studied one or few genes at a time. With whole genome sequences and new high throughput technologies, we can approach the question systematically and on a much broader scale. True. Scientists can study all the genes in a genome. For example, all the transcripts in a particular tissue or organ or a tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. That is true. A vector used to clone DNA fragments in Escherichia coli is called bacterial artificial chromosome, BAC. Chromosome constructed from yeast DNA, and it is a vector used to clone large DNA fragments. Yeast artificial chromosome. The effort to determine the 3D structures of a large number of proteins using both experimental techniques and computer stimulations is called structural genomics. Effort determine 3D structures of a large number of protein using both experimental techniques and computer stimulations called structural genomics. Determination of the order of nucleotide base sequences in a DNA or RNA molecule is called sequencing. Short DNA sequences that has a single occurrence in the human genome and whose location and base sequences are known. Such places of genome are called sequence tagged sites. Places are called sequence tagged sites. Sequence tagged sites